Buddy. Excuse me. Truth be told, not every treatment would go better if a gel were used. But I think this conference clearly shows that as we learn to employ and manipulate gels, they're offering new and important ways of dealing with difficult situations and also helping us deal with the problems of toxic and environment-ruining solvents. And really, when nothing else will work, gels are offering opportunities. This talk that I'm going to give is a small contribution to the development of use of gels in terms of grime removal. And I'll be talking about using the polyvinyl alcohol acetate borate gels that Laura described, so that'll take some time off my talk, to swab cleaning. And this is not for removal of varnish, but to look at the surfaces, and I'll show, you'll see that there is an advantage to using these gels compared to swabs for this purpose. Oops. My two co-authors on this are Teresa Duncan and Dick Weiss. I'm so sorry that Teresa can't be here today. She would have loved to have come. She's a PhD student, the way Laura was, in Dick Weiss's lab at Georgetown University. Laura's pictured here at the TechNAR conference. I wasn't able to get a picture of her at the reception on Monday night, so if anybody has a picture, please send it to me so I can add it to the collection. But the other people who have been involved in helping develop my knowledge about the polyvinyl alcohol acetate gels are Emiliano Caretti and Sheila Grassi, who come from the Luigi Dei Piero Baglioni laboratory at the University of Florence. And it's really been an interesting trip to go along and learn more about these with them. We also have to thank the people who have helped us all along the way, all the members of the Weiss group in the chemistry department at Georgetown University, and Dr. Zhang in the physics department, who helped with the AFM measurements that I'll show coming up. At the National Gallery of Art, I'm very grateful to all my colleagues past and present in the treatment labs and in the scientific research department. The funding for this project for Teresa and Dick was from the National Science Foundation, which is an unlikely supporter of cultural heritage science, so we're very grateful for that. And Teresa thanks the ARCS Foundation for a stipend. And the Curari Company, I have to stand closer, and the Curari Company generously donated the acetate gels that has already been described by Laura. And Laura already showed this picture that shows some of the advantages or properties of the polyvinyl alcohol acetate gels that make them useful and interesting for applications in treating cultural heritage objects. And most of these properties have been discussed at length for not only these gels, but other gels during this conference. And the one that I think is most interesting is the restriction of the flow of solvent. That's from a chemical point of view. But from a physical point of view, the polyvinyl alcohol acetate borate gels offer something that's very soft and, as you saw, very malleable, and shown here in this photograph that comes from the Studies in Conservation paper. They're peelable, so they can be lifted, and their properties are to some extent tunable. Both the physical properties, which can be adjusted by changing the relative proportions of the gelator and the cross-linker, and also the types of solvents that are used. And the type of solvent also adjusts the chemical properties of these gels, giving them different properties, but also materials can be added, such as surfactants, co-solvents, and actually a brand new paper that's coming out in print in November of magnetic adsorbents for cerium ions. And that brings me to a point that while we're also struggling with cleaning surfaces, other communities are too, and that paper is about removing radioactive cerium from surfaces. So we can expand our observations and our experiences because the kinds of problems we're trying to deal with are also being looked at by people in other fields. Laura has already gone through the properties of the borate gels, so I can do this really quickly. There's our hydroxyl ions replacing our acetates, which confer water solubility on the polyvinyl acetate polymer. The labile borate crosslinks that give fleeting structure to the gel that allow it to be malleable and soft. And within that network, we can have surfactants added, which form micelles in these pores, which are not constant, but actually incredibly dynamic in this particular system. We know that from recent work using single-sided NMR. For this particular study, we looked at the addition of two separate surfactants, both polyethylene glycol-based, one smaller, shorter chain length and one rather larger, the Berrie S100. The results of that have been published recently in Chem Phys Chem, a paper that Teresa is the lead author on. I will briefly show you some of the results for that application here, but as a sort of signpost, I'll say that we found that the short-chain polyethylene glycol surfactants did not seem to increase the efficacy of the gel, so the engineer's mantra KISS is operative here. We can keep the system very simple. So this talk is about removing grime from surfaces, not removing varnishes or old restorations. And the grime that we picked is the one that's been discussed already here at the conference, that was published in the paper that Bronwyn and colleagues published some time ago. It contains particulate matter, soluble particulate matter, and grease. Oops. We applied that to two different kinds of materials, brass coupons and acrylic paint. This shows the before and after on your right of the brass coupon with then the very dirty surface. It took 5 applications of the gel to get most of the grime off. Yesterday's talk that Robin gave mentioned about how tedious that can be to go through these processes, but it certainly took a lot of time, but it was effective. This is the AFM results. What you can see here on your left are the swab clean surfaces, and on the that are dry, and on the right, yes, the wet cleaning. Dry rubbing, as you might expect, left quite deep scratches in the surface. Dry rolling also left scratches on the surface, but not as deep as rubbing. Interestingly, wet rolling was not better at preserving the pristine surface than dry rolling, but the gel was able to lift the grime off without creating any scratches at all. We used a similar gel for the acrylic paint surface. It was applied, left for a few minutes, and then swab cleared, and then a small piece of that test sample was cut out and looked at with scanning electron microscopy. At the top of the columns, we have the paint before applying soil and after applying soil, and then below that in the 2 columns, the swab cleaning on your left-hand column and the gel cleaning on the right-hand column. The dirt goes into the interstices of the nubs. Interestingly, swab cleaning and gel cleaning with just plain water look rather similar, and with the addition of the surfactant, the short chain surfactant did not seem to have a beneficial effect, but the longer chain Berrie 100 in the gel gave a much better cleaning effect than swab cleaning with that same surfactant. The SEM pictures of this show the amount of soil that's removed from these. I won't have time to go through this, but fortunately it's in the book, so you can see it. The chlorine in this SEM comes from the paint, and you can see that Again, if you look at the bottom two comparisons, that the gel cleaning has removed more dirt than swab cleaning and left the surface in pretty good condition. What you can't see at this magnification is that there are little pores all over the surface. We saw more of those in the swab cleaning, so Teresa thought that perhaps that was due to re-imbibing of the grime on the surface into the acrylic paint. But since then, she did another experiment, the results of which are shown here, where she took a swab, wet swab, rolled it over the surface of an unsoiled acrylic paint, and on the same paint used the gel, and on the swab-treated surface, there are many, many, many pinholes, which are probably air holes in the acrylic paint. But what it means is that swab cleaning took off the very top surface of the acrylic paint, whereas gel cleaning preserved that surface very well. Now, that's not to say that in the clearing step that wouldn't happen, but at least in the first step we can see that swab cleaning is a little harsher to surfaces than gel cleaning. So, in conclusion, the gels provide a very gentle way to treat a variety of chemically and physically sensitive surfaces, mitigating the formation of scratches. We looked at brass, but imagine looking at gold foil or vermeil, for example. So very thin surfaces would be beneficially treated using these gels. The development of pinholes, which we only probably exist all along, but become visible on an acrylic surface when the very top skin is removed. And finally, the addition of short chain surfactants does not seem to be worth it, and a small amount of the longer chain polyethylene glycol is effective at lifting grease. Thank you.